All right, welcome to part 3. In here, we will take the DNA sequence that we have trimmed from the part 2, and we're gonna plug it in to the DNA sequence alignment software, to our DNA sequence alignment tool. We call it BLAST, and hopefully we're gonna get some result, and we're gonna talk about what we see over there. Okay, so let's go. We're gonna go to BLAST. So how do we find BLAST? Well, you can ask Dr. Google. Okay, go to Google and then just type BLAST, B-L-A-S-T. What? B-L-A-S-T, something like that. No, BLAST from the past. What? Okay, just go there, BLAST, and then you go to the site maintained by NCBI. Just go there. By the way, NCBI is National Center for Biotechnology Information. This is a subset of project maintained by the NLM, National Library of Medicine, which received funding from NIH. Here what you see, this is the BLAST that you can use. There are several versions of BLAST. In this video, I'm not gonna go through what's the difference between different kind of BLAST. There's BLAST N for nucleotide, there's BLAST X, T plus N, and BLAST P. Of course, there is a T plus X as well. So what we're gonna use in here is this so-called nucleotide BLAST or BLAST N. Okay, so you just click on this. That here's the BLAST N, BLAST P, T plus X, T plus N, and T plus X over here, right? Okay, so here is where you put in your DNA sequence. So where's our DNA sequence last time that we trimmed? Right here. So this is your DNA sequence, right? So now we want to put it there, but before we put it into the box in here, you have to put it into this so-called FASTA or FASTA. Some people call it FASTA sequence format. Okay, the FASTA format or FASTA format typically start with the initial line number 1 as the title, if you will. And the way you tell the algorithm that this is a title, you have to give what we call caret sign. Right here, caret sign, and you can name your sequence anything. Okay, let's just call this sequence, I don't know, trial by fire. Okay, trial sequence. You can name it anything. You can name it AK105. You can name it McDonald, or you can name it anything. So here I just call it trial, and then just copy everything, copy that, and then put it into this box right here. So by having this caret sign, it tells this algorithm whatever come after the trial sign or after this caret sign is the name, okay, of your query. Okay, so you can do several stuff. You can upload a file if you want, or you can use this so-called accession number, which I'm not gonna talk about it in here, or GI and so on and so forth. But here let's just do a very basic sequence analysis right here. Okay, and what you need, okay, so here's what you want to align it with. Okay, now by default you are aligning your sequence to any available sequence in the database, in the standard database. This is the NR, simply stand for non-redundant database. For us, we know this is a sequence from the ribosomal RNA, so we want to use this particular database, right? You click on that. You can use that if you want standard database. You can use that. It's just you will end up with a lot of sequence coming from the metagenomic sequencing result. A lot of them are uncultured, because if you do metagenomic, you simply take environmental sample and you sequence anything that's in that environmental sample. Some of them you have not cultured it yet, so that's oftentimes is late. Those kind of sample with no culture, they are simply labeled uncultured bacterial species X or something like that. So if you do this, you can, but you will end up with a lot of those uncultured bacterial sample, right? So here we want to align our sequence only to the one that we has been cultured, okay, and we know what that is. Okay, so here, once you do that, you can choose what you want. Here for us, it's gonna be 16S ribosomal RNA because our sample is bacteria, or this one can be used for archaea as well. But if you're using fungi, for example, then you use the 18S rRNA, or you can use the 28S for the fungi as well, or the ITS, the internal transcript spacer. So that's what if you're working with fungi. Okay, now for us, we're doing this 16S here for bacterial and archaea. If you sort of have idea what your organism is, you can actually type it here, like Staphylococcus. You can type it here, Staphylococcus, and they have this taxonomy ID over here. By doing, you can either you can do this like this, then it will only search your sequence. It will only align your sequence against the staphylo or member of this Staphylococcaceae in a database that belong any member of this this group within the available sequence in the database. Okay, or you can decide to exclude them. You can do that too. If you want to exclude this particular one but not other, you can certainly do that. So there are a lot of things you can do, but for us, we want to do we're not going to include any organism here. We want to do unbiased alignment to any available sequence in the database. Okay, so that's why we don't use anything in here. Okay, don't worry about this for now. This is for more specific. Here's the uncultured, exclude uncultured. You can do exclude uncultured if you want. You can still use standard database and then exclude the uncultured if you want. You can do that one too. But here I am using only the database for the 16S ribosomal RNA. Okay. Now don't worry, there are other parameter that you can choose here. We can certainly talk about that later in the other video if you are interested. But for now, let's just do the basic blast and see what they are. Okay, here I usually like to show the result in a new window. That's what my preference, so that way I can reuse the same window and then just simply switch up the query sequence right here. Okay, so because if you click plus over here, it goes to the other sequence here, and it takes some time. It takes a few seconds to a few minutes. While doing so, you can do another sequence. You can put another sequence and let it run another window, and you can run multiple jobs at the same time. Right, that way you save time. So that's one that's what my preference is. Okay, so see, it's actually not that bad, not too long. So because ours is pretty simple and straightforward. So here, this is the alignment result that you get. Job title trial. That's the title you put, and then here's just the summary of what you have. The query length is 510 right here. That's just the length of your sequence. Okay, so here's the result that you get. So you have the name of that particular organism. It looks like our organism is not Staphylococcus epidermidis. The top hit is Staphylococcus edaficus, several Staphylococcus saprophyticus right here, Staphylococcus xylosus. They're actually pretty close together, but very far from Staph epidermidis. So where is our Staph epidermidis? We can take a look at all the way down here. Equorum. Here. This is your Staphylococcus epidermidis, all the way down here. Yeah. So it's not. It's rather different. Actually, quite distant from the sequence that we get from our sequencing result, right? So now, if you get something like this, how do you know that? Yeah, well, I told you the top one usually is the correct one. So one way to, if you don't really care about validating your result, then you just take the top one, and then here, this is mine unknown, and you're done. Okay, that's pretty much what you can do. But here in this lecture series, I'm going to show you sort of like what are some of the numbers, what these numbers mean, and what sort of what do they represent, and how can you be very confident that this is truly your the identity of your organism. Okay, so there are several things that you can see here. I'm not going to talk about the max score and the total score. That's in summary, that's essentially is when you're aligning your query sequence versus the one in the database. Every time there is a match, letter that match, let's say in your query you have A, and then in your database in the same position you also get A, you get some point, positive point, +1 or +2, something like that. And every time there is a mismatch, you get penalty, -2 or -3. And if there's a gap, when there's a gap, you also get penalty, right? So no, we're not gonna go through it in here, but essentially you add all those scores together, and that's the score you see over here. The query coverage is 100%. It means the sequence completely match with what the sequence in the database is. 100% match. Now, the most important number in here is arguably is the e-value right here. The e-value is a statistical number that tells you how likely is it for you to get this hit by chance. Essentially, if you have a large e-value number, it means the chance of you getting this particular match by random is pretty high. So you don't want high e-value, you want low e-value. So the lower the e-value, the better, the higher your confidence that this match is actually real. But if you look at all this, look at that, almost everything have very low e-value. It's just essentially 0, right? So if you go all the way down, it's still 0. So they're very, very closely related, this particular alignment between your query and all these other organisms in the database. Even the one that belong to different genus, for example, Bacillus right here. See, Bacillus, the e-value is still 0, right? Everything is still 0, right? Although they're slightly different in terms of the coverage, and here the person identity. The person identity essentially is like once you align them, let's say you get 100% coverage within those that you align, what's the percentage of match? If it's 100%, it means all of the letter in the sequence match. That's what 100% match is. So if it's 99.8, you may have 1 or 2 mismatch. Things like that happen. It can be because your sequencing, resolution is not great. It's possible, right? So 100% identity is usually a bit rare. So anyway, so yeah. Okay, again, if you look at this, almost everything match really well, right? Now the question is, how do I know that my organism is Staphylococcus edaficus, for example, and it's not Saprophyticus? After all, the e-value is very closely related, max score is really closely related, right? Or even, how do I know it's not Staphylococcus psilosus, right? The e-value is the same, person identity is slightly off, perhaps there are more mismatch over here, but everything else is the same. Query coverage is 100%, max score is slightly lower. But how do I know that my organism is truly belong to, let's say, Staph edaficus or Staph saprophyticus, and not, let's say, Staphylococcus hominis, for example, right? How do I know that? Well, in order to answer that, you can take a look at this so-called the MSA viewer like this. Okay, so in fact, in the last video, we're gonna check them out together. But yeah, I think that's pretty much. I'm gonna stop here for part 3, and we'll look at the MSA here, which is the what we call multiple sequence alignment, and from there we're gonna analyze how well this actually match between your query to each of this sequence that you have over here. Okay, all right, so I'll see you in the part